Hello everyone, my name is Dr. Ambud Misra, and in this video we are going to discuss about lipid storage disorders, that is your sphingolipidosis. But before that, let's have a better understanding about the substrate and the enzymes and their defects that are going to contribute to these diseases. So starting with the phospholipid. So phospholipid is a kind of lipid where there are four components: your fatty acid, alcohol, phosphoric acid, and your head group. So this alcohol, this is going to form your backbone. Means other components are going to attach on this alcohol, on the chain of this alcohol. So this alcohol is going to form the backbone. And other than that, we have fatty acid that is going to get attached on this alcohol, and the phosphoric acid also is going to attach on this alcohol, and this head group is going to in turn attach to this phosphate group. So this head group can be of two types. One is your nitrogen base containing, and another one is your non-nitrogen base containing. So one base where there will be nitrogen and another one where there will be no nitrogen. So there are two kinds of head groups that are going to attach on this phosphate group, which in turn is going to attach on this alcohol backbone. Now there are two types of phospholipids, that is your glycerophospholipids and your sphingоphospholipids. So what are the differences between these two kinds of phospholipids that we are going to see? So in glycerophospholipids, the backbone, the alcohol that is your backbone, which is going to be formed in case of glycerophospholipid, is your glycerol. That is obvious by the name. It's glycerol. But in case of sphingophospholipids, the backbone is going to form by sphingosine. This is your sphingosine. Sphingosine is nothing but it is 18 carbon amino alcohol. This is also an alcohol which is your 18 carbon amino alcohol. So the backbone in case of glycerophospholipid is your glycerol, and in case of sphingophospholipid is your sphingosine. Now the number of fatty acids that are going to get attached to this glycerol is 2, whereas in case of sphingophospholipid, the number of fatty acid that is going to attach on this sphingosine backbone is your 1. So these are the two major differences between glycerophospholipid and your sphingophospholipid. Backbone is your glycerol. Here it is your sphingosine. There are total number of fatty acid is your 2, and here it is your only 1 fatty acid that is going to attach. So this is the major difference between glycero and sphingophospholipid. Now we will see the examples of glycerophospholipid. One is your nitrogen containing base and another is your non-nitrogen containing base. So in case of nitrogen containing base, we need to remember these examples. That is your lecithin, your cephalin, and your phosphatidyl serine. These are your nitrogen containing bases. The glycerophospholipids where the base is nitrogen containing. The examples are lecithin, cephalin, and your phosphatidyl serine. Now in case of non-nitrogen containing bases, the head groups that are non-nitrogen containing, the examples are phosphatidyl glycerol, this is your phosphatidyl inositol, and diphosphatidyl glycerol. These are your non-nitrogen containing. Now the example of sphingophospholipid, the major example, the only one that you need to remember, which is really important, is your sphingomyelin. Sphingomyelin. Sphingomyelin is your sphingosine containing, lipid. But glycerophospholipids are not sphingosine containing lipids. So when you say sphingolipidosis, it does not involves the fatty acid which does not contain sphingosine, that is your glycerophospholipids. These does not contribute, these does not going to form any disease which are included under the sphingolipidosis. So it's only sphingomyelin and the defects related to sphingomyelin that you are going to see in sphingolipidosis. So this is the basic about phospholipid. You see what are the components, that is your fatty acid, alcohol, phosphoric acid, and the head group. Based on head groups we saw the examples, and the main example that you need to remember in case of sphingolipid is your sphingomyelin. The differences between glycero and sphingolipid is your the backbone is glycerol in glycerophospholipid, and in sphingophospholipid it is your sphingosine that is going to form your backbone, and the number of fatty acids 2 and 1 respectively. Now we will see the basic structure in order for better understanding of glycerophospholipid. So you see this over here is your glycerol component, these two are your fatty acid component, this is your phosphate group, and this is your head group. See, the fatty acid is directly attached to glycerol backbone. Phosphate is also directly attached to glycerol, but this head group, it is attached to this phosphate, not it is attached directly to the phosphate group. So this is your phosphate ester linkage. So your head group is attached to phosphate via phosphate ester linkage. Now one more thing that you need to remember here is that what are the enzymes that are going to break some specific bonds at specific locations. So if in order to break this bond between the glycerol backbone and the first fatty acid is your phospholipase. This is your phospholipase A1, and the bond between the see when the phospholipase A1 is going to be acted on the whole glycerophospholipid, the first the phospholipase A1, the bond that is going to break is going to break the fatty acid attached to alpha carbon of the glycerol, the first fatty acid, this one. And phospholipase A2 is going to break the another fatty acid that is attached to glycerol backbone, that is at the beta carbon. So phospholipase A1, the first fatty acid, phospholipase A2 is going to remove the second fatty acid. Now there is phospholipase C. This is your phospholipase C that is going to break the bond between the glycerol backbone and phosphate, and here phospholipase D is going to remove the head group from the remaining components. So these are the enzymes which are going to act on the specific location of glycerophospholipid, and in turn is going to remove some specific components. So this you need to remember. Now what is the basic structure of sphingophospholipid? So in sphingophospholipids, the components that you are going to see is your sphingosine, which is your backbone. Then you have your phosphate group. Then you have choline and your fatty acid. So the choline is going to be a head group here, and these two combined is known as phosphoryl choline. So what is remaining here? The remaining component is your sphingosine and fatty acid, which is, which combined is known as your ceramide. It is known as ceramide. This is your ceramide. So ceramide and phosphoryl choline, when these two components are going to combine, this is going to form your sphingophospholipid. And further, if you have to break down, ceramide, it is formed by two components, that is your sphingosine and fatty acid, and phosphate group and choline, that is your head group, which is a base. These two combined are known as phosphoryl choline. So these terms, this ceramide, you really need to remember. And see, now we'll see what is phosphatidic acid. This we are going to see again. So what is phosphatidic acid, or you can say phosphatidyl group? See, in if in glycerophospholipid you remove the head group, and the remaining components that are left behind is known as your phosphatidic acid or phosphatidyl group. So what are components that you are going to see in phosphatidic acid? That is your glycerol plus fatty acid plus your phosphate. These three combined is known as phosphatidic acid, and ceramide, that is your sphingosine plus fatty acid. So these two, you need to remember that if we remove the head group from glycerophospholipid, what left behind is your phosphatidic acid or phosphatidyl group. That is your, the components are glycerol, fatty acid, and phosphate. And ceramide, the components are sphingosine and fatty acid. So these four are the components for your sphingophospholipid. And if we remove the phosphate, this phosphatidyl choline thing, the what is left behind is your ceramide, that is your sphingosine plus fatty acid. Now you see the head group, it is attached to, it is attached to your phosphate group via phosphate ester linkage, see, which in turn is attached to the alcohol backbone. Now we will see some these, we will further see these individual glycerophospholipids, and we will see how they are formed and where are they found inside the body. So phosphatidyl choline, which is also known as lecithin, which when the phosphatidic acid, this phosphatidic acid, if you remove the head group, so in turn we are going to see which head group is going to combine to phosphatidic acid in order to form lecithin or phosphatidyl choline. So from the name, it is obvious that if you combine phosphatidic acid with choline, it is going to form phosphatidyl choline or lecithin. Now where you are going to see lecithin inside the body and what are the functions. See, it is lecithin, it is your most abundant, this is most abundant phospholipid in cell membrane. Inside the cell membranes, this is the most abundant phospholipid. It is also the storehouse of choline. It also forms mucus layer. It forms mucus layer, and it is the major constituent of your mucus layer. Now further, it also has emulsifying property. It also has emulsifying property. And because of this, it is going to break down fat and also decreases low density lipoproteins and increases high density lipoproteins. These are good one, this is the bad one. High density lipoprotein is a, it is said to be good, and whereas the low density lipoprotein is said to be bad. So this is the function of, these are the function and the location where they are found. That is your lecithin or phosphatidylcholine. Next one is your phosphatidylethanolamin, that is your cephalin. So which base group is going to combine to phosphatidic acid in order to form cephalin or phosphatidylethanolamin is obvious, it is your ethanolamin. Now where are these found and what are the functions? So the functions of cephalin, this you need to remember along with these names. So the function is your blood coagulation. It maintains cell membrane structure, and it also has function in brain and neural function. Brain and neural structure. These also maintain the neuronal and brain structure. Now we saw cephalin, which the base group is your ethanolamine. It is important for blood coagulation. See, these glycerophospholipids, these most of them are found in the cell membrane, plasma membrane. So by one way or other, they all have some functions that is going to maintain the fluidity or structure of the plasma membrane. But the most predominant is your lecithin, which is also found in mucus layer. This you need to remember. Most abundant phospholipid in cell membrane is your lecithin, that is your phosphatidylcholine. It is also found in mucus layer. And your cephalin, this is also found in cell membrane, which is going to maintain its structure, and it helps in blood coagulation. The next one is your phosphatidylserine, and it is going to form by the base group serine. It's obvious by the name. And the function is, it is mediator of programmed cell death, or you can say apoptosis. This is apoptosis. So phosphatidylserine is involved in the process of apoptosis. Moving on to the next, your phosphatidylglycerol, which is your non-nitrogen containing base. And the head group in this case is going to be your glycerol, obvious by the name. And see, phosphatidylglycerol, this is also maintain the structure of cell membrane or plasma membrane, and this is also involved in the synthesis of cardiolipin. Now we will see further what is cardiolipin. Next one is your phosphatidylinositol. In this, the head group is your inositol, and the function is that it is the secondary messenger in hormonal pathway. You see, you have seen certain hormones that are going to reach the target or reach the nucleus by through the second messenger, and that is your phosphatidylinositol, sometimes abbreviated as IP3. You must have seen this in uh, how like hormones act when they go to extracellular receptors, and they release the secondary messengers that are going to reach to the nucleus in order to further release certain proteins or certain enzymes. So this is your phosphatidyl inositol, and it is important that it is the secondary messenger in hormonal pathway. The next is your diphosphatidylglycerol, glycerol, which is also known as cardiolipin. So what happens here, the base group itself is your, it's like there are two phosphatidyl groups. See, one phosphatidyl group and another one, and these two are linked together with the glycerol component of glycerol in between. So you can say a phosphatidyl group with a glycerol linked to it, when it is going to attach to the phosphatidyl group, is going to form cardiolipin, that is your diphosphatidyl glycerol. So this the function of cardiolipin, it is that it is only antigenic phospholipid. It has antigenic properties, and it is the only antigenic phospholipid that you are going to see. The next function is that it is important for mitochondrial function and electron transport chain. Mitochondrial function and electron transport chain. So by the name cardiolipin, you may wonder that it must be present inside the heart, but it is not so. It is present inside the heart, but like mainly it is present inside the mitochondria, and it is responsible for the functioning of mitochondria and electron transport chain, and it is the only antigenic phospholipid that you need to know. Now moving on to sphingomyelin. So sphingomyelin, it is present in the outer plasma membrane. Most importantly in myelin sheath. This is the phospholipid that is going to form myelin sheath. And the next function is that it is going to form lipid rafts with cholesterol. So sphingomyelin is going to form lipid rafts. What are lipid rafts? Lipid rafts, these are structures that is present in the plasma membrane, and through which you will see the cell signaling is going to happen. And some pathogens, this is also going to form the entry pathway for some pathogens. So lipid rafts, first it is for cell signaling, and the second, it is going to be entry point for some pathogens. To enter inside the cell. So sphingomyelin, the main function that you need to remember, it is responsible for forming the myelin sheath, and it also forms lipid rafts that is responsible for cell signaling and entry of certain pathogens. Now we will see degradation of glycerophospholipids, how these glycerophospholipids are going to get degraded, and it is done by certain enzymes. So we saw previously that it is your phospholipase A1, phospholipase A2, phospholipase C, and your phospholipase D. This is the PLP stands for. phospholipase. Phospholipase. Now, phosphatidyl inositol, when it is going to act on pi phospholipase A2, it is going to form arachidonic acid. Now, this arachidonic acid is going to be substrate. It is going to form the substrate. It is going to be the substrate for synthesis of prostaglandins. See, in the previous lecture, we saw that arachidonic acid is going to be the substrate for formation of certain certain prostaglandins, leukotrienes, and some cell mediators. So it is so the substrate for for formation of arachidonic acid is your phosphatidyl inositol, which whenever it is going to be acted by phospholipase A2 is going to form arachidonic acid, which is going to be the substrate for synthesis of prostaglandins. Now, certain places where phospholipase A2 is found is your snake venom, your bee venom, and your pancreatic juice. Now moving on to degradation of sphingomyelin, how it is being broken down. That is your sphingomyelin when it is being acted upon by sphingomyelinase. Sphingomyelinase is going to form ceramide, and when ceramide is being acted upon by ceramidase, is going to form sphingosine and fatty acid. Now, this sphingomyelinase and ceramidase, the deficiency of each of them is going to contribute to development of certain diseases that we will see further. Now moving on to, but you need to remember these, that sphingomyelin is broken down to ceramide with the help of sphingomyelinase, and ceramide is broken down to sphingomyelin and free fatty acid through ceramidase. It's easy to remember because it is based on the name of their subsequent substrate. Now moving on to the next, that is your glycolipids. So how what are the components which are going to form glycolipids? So that is your sphingosines plus fatty acid and sugar. Any sugar like monosaccharide, disaccharide, oligosaccharide. So when the sugar is going to combine with sphingosine and fatty acid, that is your ceramide that we already know. So whenever a sugar component is going to get combined with the ceramide, is going to form glycolipid. One important thing that you need to remember, that here you are not going to see any phosphate. It is without the phosphate. And we already know that sphingosine plus fatty acid is your ceramide. Now, what are the types of glycolipids? We need to remember the majorly three types of glycolipids. That is your cerebroside, globoside, and your gangliosides. So how the cerebrosides are going to form? See, your glycolipids are going to form when a sugar component is going to attach to the ceramide. So what sugar is going to attach to ceramide so that it forms cerebroside? That is your monosaccharide. When a monosaccharide group is going to attach to ceramide, it is going to form cerebroside. Further, the cerebrosides are divided into two types. One is your glucocerebrosides and another one is your galactocerebrosides. So in case of glucocerebroside, your monosaccharide is going to be glucose, and in case of galactocerebrosides, your monosaccharide is going to be your galactose. And where these are found is your glucocerebrosides are found in non-neural tissues, non-neural tissues, and your galactocerebroside is found in neural tissues. That you need to remember. The galactocerebrosides are found in neural tissues, whereas glucocerebrosides are found in non-neural tissues. So that means if any enzyme which is responsible for breaking down galactocerebroside is defective, this is in turn going to, defect. The defect that you are going to see will be in neural tissues. Like you will see something related to, your neural function, that is your mental retardation, or your delay in the development of your brain, and etc. Another one is your globocytes, where the ceramide, when it is going to combine with disaccharide or oligosaccharide, is going to form ceramide. One example is your lactosyl ceramide. Now lactosyl ceramide, it is present in erythrocyte plasma membrane, and here this here the disaccharide is your lactose. Here the disaccharide is your lactose. Another one is your ganglioside. So how the gangliosides are formed? Whenever the ceramide is going to get combined with oligosaccharide. One example is your this. What is this? What this stands for? That is your N-acetyl neuraminic acid, or this is also known as sialic acid. This is sialic acid. And gangliosides are mainly going to form the components of neural tissue. So use a neural tissue here and use a neural tissue here in galactocerebrosides and in case of gangliosides. Now let's see some very important enzymes and substrates, that are going to contribute to certain diseases, that is your sphingolipidosis, when they are defective. So see, gangliosides are of certain types. It's named like GM1, GM2, GM3, like GM1, GM2, GM3. These are, they have got this nomenclature based on, this G stands for your ganglioside, M is your monocylo, containing, and 1, 2, 3 is your based on your chromatography. Now GM1 gangliosides are going to convert into GM2 ganglioside by the enzyme that is your beta galactosidase, and your GM2 is going to convert into GM3 ganglioside with the help of beta hexosaminidase, which has two isoforms. It has two isoforms, A and B. It has one alpha and one beta subunit, and it has two beta subunits. So we will see when these different isoforms are being defective, and then what disease are they going to contribute to. So GM1 is going to convert into GM2 with the help of beta galactosidase. Remember, this is your galacto, not gluco. And GM2 is going to get converted into GM3 ganglioside with the help of beta hexosaminidase. Now, glucocerebrosidase is going to convert into ceramide plus glucose. You know, the glucocerebrosides are formed by combining ceramide with glucose. So when the glucocerebrosides are, broken down, these are going to form. This is going to form ceramide plus glucose, and this is done with the help of enzyme that is your beta glucosidase. Beta glucosidase. Here you need to remember it is glucosidase, not galactosidase, because beta galactosidase is you are going to see in case of GM1 ganglioside, and in case of glucocerebroside you are going to see beta glucosidase. And what about galactocerebrosidase? When galactocerebrosidase are going to be broken down into ceramide plus galactose, the enzyme involved is your beta galactosidase. So beta galactosidase has two functions, that it is going to break down GM1 ganglioside, and it is also going to break down galactocerebrosidase. So galactocerebrosides. Now you see sphingomyelin, when it is going to be broken down into ceramide plus phosphorylcholine. The enzyme is your sphingomyelinase. And when ceramide is being broken down into sphingosine and free fatty acid, it is your ceramidase. So these enzymes you need to remember. And further, when globotriacyl ceramide is going to broken down into ceramide plus triglycoside, this is your globoside. This is an example of globoside, and this is going to break down into ceramide plus triglycoside with the help of an enzyme known as alpha galactosidase. So we need to be careful in these. Here, beta galactosidase is also going to break down GM1 ganglioside along with galactocerebrosidase. So like from galacto, you can remember this galacto, and from gluco, you can remember this gluco. So beta glucosidase is going to break down glucocerebrosidase, and beta galactosidase is going to break down galactocerebrosides, and your beta galactosidase is also going to break down GM1 ganglioside, and your alpha galactosidase is going to break down this compound, which is your globoside. Now this was all about the enzymes and the substrates that are going to involve in diseases which is your sphingolipidosis. So if you remember these enzymes and substrates, it will be a lot easier for you to understand those disease particular. So there are around 7 to 8 diseases that we are going to see. So these enzymes, when are defective, are going to accumulate the substrates that these enzymes are going to break down. These substrates are going to get accumulated in either inside the lysosome or inside the cell. In turn, these substrates are going to kill the cell, and which in turn is going to contribute to the formation of symptoms of that particular disease.